warm welcome to today's Minds, Technology, and Society seminar, and want to thank the Leszko Samuelson Fund for sponsoring this seminar. And today's speaker, Dr. Heather Dean, joins us from the U.S. Department of Food and Drug Administration. And Heather has actually had an interesting career path, and she'll be speaking more about this in greater detail in the body of her talk. But just to give you a couple of salient tidbits, she was previously with NSF, I'm sorry. And also before that, she was a postdoc at the Center for Neurosciences at New York University. And on a purely selfish personal note, this is a kind of a special occasion for me. All of our MTS speakers are absolutely great, and I can say that because I picked them. But this is an unusual and personally special occasion for me, because this is the first MTS speaker that we have this semester. It's that who so happens I was able to attend the commencement ceremonies for both of Dr. Dean's degrees, her undergraduate degree at California Institute of Technology and her doctorate at Duke University. And so with that, let's give Dr. Dean a warm welcome.
You didn't mention it, but he was one of my very first professors, so we've known each other since I was 18 years old. So, as Peter mentioned, I am now at the Food and Drug Administration, and I'll talk about that a little bit at the end. Peter asked me to talk about the science that I did when I was at NYU as a postdoc. So this was all work that was done 5 to 8 years ago. So at the very end, I'm going to tell you a little bit about what I've done since I went to Washington, D.C., both at the National Science Foundation, through the American Association for the Advancement of Science, a little bit of work I did as a consultant for the BRAIN Initiative, and then the work that I'm doing at FDA, so that if you have any questions, you can ask about that as well as about the science. Because this is a class about minds, technology, and society, and I'm very interested in the science policy side of things, the society part. But for the science talk, today I'm going to talk to you about understanding the neural basis of hand-eye coordination. So first, we think about how might the brain coordinate movements, and how can we study this? So I get at this with a combination of looking at the behavior and finding a behavioral signature of coordination, using mathematical models then to look at what might underlie that behavioral signature of coordination, and then actually going into the brain and looking for signals that might help us verify some of these things that we looked at with the mathematical model. So first of all, it's long been known that when movements are coordinated, such as the saccade and reach, the reaction times are correlated. This is actually data from one of my monkeys, Jug, Monkey J, I'll refer to him here. And it's a single session, but it's very representative of the sessions that I had with all of my monkeys. Let's see how it... So you'll notice here, I've plotted the reaction time for the saccade, or eye movement, against the reaction time for the reach. And you'll notice that when he's faster to make the saccade, he tends to be faster to make the reach. When he's slower to make the saccade, he tends to be slower to make the reach. There might be several potential explanations for this that have nothing to do with coordination. You might think there could be a global signal. He might be more motivated on some trials than others. These monkeys work for juice. They come into the lab thirsty. He might be more motivated early in the trials and satiated later. There could be other explanations, such as bottleneck effects. So I'm going to go into how we looked at this. We wanted to tease apart these two movements in time and kind of vary the degree of coordination, which I think of as overlaps in the plans. So we do this with a task that we call the stimulus onset asynchrony task. And I'm going to walk you through this figure because it's a little complicated. So these are highly trained monkeys. They're trained to touch green squares on the screen and look at red squares on the screen. And then once they do that, a target appears. In this case, it's a yellow target, which is signaling to him that he's going to move both hand and eye to that target. And it appears in one of 8 locations around that central touch and fixation. As soon as that target appears in this task, the red fixation also disappears. That's his cue to move his eye then to the target. Most trials in this task, we actually have him simultaneously cued to make the reach, but in some trials, we tease this apart with a stimulus onset asynchrony. This is an SOA that's variable in time from 1 to 620 milliseconds. After that SOA, he receives an auditory cue to move his hand to the target for his reward. And we use an auditory cue here. We could have just as easily used a visual cue, but as many of you probably know, when the eye is in motion, there's no visual processing, and that would affect our measurement of reaction time. So we use an auditory cue here. So we can then look at how this SOA, this difference in timing between the go cues for each of these movements, affects either the reaction time or the correlation in the reaction time that I showed you later with that sample. So looked at this in 3 monkeys. First here, I'm showing you the saccade reaction time as a function of that SOA. So I've binned these trials according to the length of that stimulus onset asynchrony for these 3 monkeys. And what you'll notice is that the saccade tends to be faster when the SOA is shorter for all 3 monkeys. And then, especially once the SOA is longer than the saccade reaction time, so the reach is being cued after that saccade, then the saccade is slower. And this is behavioral data, so it's messy. You'll notice that monkey S, there seems to be some kind of perhaps a startle response in here when the target appears just a few milliseconds after the auditory cue appears soon after the cue to make the saccade. Again, behavioral data is messy. But in general, you see facilitation of the saccade for shorter SOAs. Here I show the reach reaction time, and there are a number of things going on here. You do tend to see some facilitation at the shorter SOAs. You also see that the monkey is faster for these longer SOAs, and the reason for that might be expectancy. So we chose SOAs that were 1 to 620 milliseconds. We chose that number because that's about twice the average saccade reaction time, because we wanted to have reaches that were cued before and after the saccade begins. But monkeys get to know a trial when they've done thousands and thousands and thousands of trials. So this monkey, if he's been waiting 600 milliseconds, he knows that cue to move is going to be coming very soon. So there might be an expectancy effect, and that's been shown in a number of different paradigms to speed reaction time. You'll also notice that for these 2 monkeys, he's slowest to make his reach when the SOA is 0, and this may be because, unlike for the longer SOAs, where he's already been given the location cue because he's making his saccade to that location first, in the 0 SOA case, he has to process that visual information before he makes his reach. So knowing where you're going to move ahead of time has also been shown to speed reaction times. And so that may be why this is longer. And you'll notice that you don't have that with monkey S, but he was much slower to make his reach in general. So there might have been more of a cushion there for that visual processing. Finally, we'll look at the reaction time correlations for the saccade and reach. And here for all 3 monkeys, you'll notice that the reaction time is greatest when the SOA is 0. So when those movements are cued together, and it drops off then as a function of increasing SOA. So now I'm going to go into the model that we use to look at, to help explain this behavioral data. I'm going to be a little bit hand-wavy here because I know there's a variety of expertise in the room. We're looking at this as a leaky accumulator. And just think of this as a group of cells. There's some level of self-excitation, and there's some input, right? And the activity in this group of cells, if the input is high enough, it's going to begin to accumulate until it reaches some threshold, and this is a noisy accumulation, and so you're going to wind up then with a variety of reaction times, and so this can help explain that variety of reaction times defined as time to threshold. And this type of model has been shown to describe behavioral data in a number of paradigms, including decision making, accumulation of evidence, and reaction time. And this has also been shown to relate to neurobiological models. So neuroscientists have gone in and found signals in the brain that seem to relate to these accumulators. So a little bit more about the input to these accumulators. We're going to imagine that there's a DC component. So this is going to be something that's 0 if the monkey is not planning to make the movement. It goes up when the cue to make that particular movement is given. And when that threshold is reached and the movement is made, it goes off. There's also a noise component that's white and Gaussian, and that gives us that variety of reaction times. We also don't want this to start accumulating simply noise, so there's some threshold for the input beyond which it begins to accumulate information. So let me show you how this might work for these two accumulators. So you have an accumulator for saccades, you have an accumulator for the reaches, and this is the input coming in, and again it's that noise plus the DC component that begins when the saccade is cued, and it accumulates until it reaches that threshold, and then the input, the DC component, is shut off. There's the variable SOA, after which the reach is cued and the reach accumulation begins. And notice that there's some overlap, and this overlap is going to depend not only on the SOA, that time between the go cues, but also on how quickly he begins his saccade, right? So for a given SOA, if he's faster to make his saccade, then there's going to be less of an overlap between the plans, and that's going to become important later. Oh, I should have said at the outset, if you have any questions, please jump in. I'd rather people stay on board than wait until the very end and have me flip back to something that they didn't understand. All right, so now I'm going to go through different models. Remember I said there could be many potential explanations for that reaction time correlation that we saw in the behavioral data I showed you. One could be global modulation. So you can think of this as arousal or motivation, and you could think of this as changing the gain. How quickly do those accumulators accumulate to threshold? You might also think of it as a change in the threshold. But let's think of this as changing the gain. And arousal effects have been shown to be mediated by cholinergic and aminergic signals from various areas of the brain. Sorry. And these are mediated on very slow timescales, hundreds of milliseconds rather than a few milliseconds. So it's not changing within the trial. And so we're going to imagine for these accumulators that the effect is the same on each of these accumulators within a trial and can vary across trials. So now we can think about how this model would affect the reaction time, both saccade reaction time and reach reaction time. It's not dependent on the SOA. We see that the reaction times are not dependent on the SOA because this is simply a change in gain. It's not going to change how quickly, it's changing how quickly there's a race to threshold, but it's not varying as a function of the SOA. We look at the correlation in reaction time as a function of the SOA, and we see that, again, there is no effect as a function of the SOA. If the changes in gain are more variable, you see a higher level of correlation. You can think of this as, for the slower trials, he's much slower for both movements, and for the faster trials, he's much faster for both movements, and so that leads to that greater level of correlation. But you do not see any change across SOA because these are the same in both the accumulators within a trial. You might also imagine that you could wind up with correlated reaction times when you have common input to these areas, right, that encode these plans. And that common input might be the noise, or it might be the DC component of the signal. So let's look first at potential for shared noise, right? You could imagine that these are areas that might be processing input from the same area. There's signal in there that's not necessarily related to the plan. We're going to imagine that some fraction goes to both. We look at reaction time as a function of the saccade onset asynchrony. That's not going to change, but correlation does. You see positive correlations in reaction time for the shorter SOAs. This is because when you have noise that, say, drives one accumulator to threshold faster, it's also going to drive the other accumulator to threshold faster if that noise is being input while both of these accumulators are being driven by the plan. And the less the plans overlap, the less correlation you're going to see in the reaction times. And then once the saccade is made, once the SOA is greater than the saccade reaction time, you're going to see zero correlation. Does that make sense? Okay. So this doesn't quite explain the behavioral data we looked at. So let's look at shared signals. You might imagine that when one movement is cued, it could influence the accumulation in the other. So here we look at some fraction of the signal being shared. We see that the greater the fraction, the more facilitation in the reaction time, because these are going to be driven faster to accumulation if, while they themselves are accumulating information during their planning, they're also receiving a signal from the other plan. However, when we look at the reaction time correlations, these are negative. And that's because for those trials that are where the saccade is driven by noise more quickly to threshold, that's going to shut off this input plan, right? I said that the input shuts off when the movement begins, and so then the reach accumulator no longer has that additional drive, and so those reaches are going to be slower than if the saccade had taken more time. And so you wind up with this anti-correlation. So none of these have quite explained the data we've seen, so we can next look at what I think of as coordination, which is this interaction between the plans themselves. So we model this as a model of mutual excitation, where activity in one accumulator can affect the activity in the other and vice versa. And you might imagine these could affect each other in the same way or in different ways. So let's look at two extremes of this. One, a symmetric mutual excitation model, and one in which the reach influences the saccade. I actually didn't give a very good introduction to this. So it makes sense in many ways that the reach could affect the saccade. We make saccades, eye movements all the time, alone without a reach. However, when we reach to pick something up, we often look first to guide our hand to the target. So it would make sense, perhaps, that the reach could affect how we move our eyes for visual guidance, for improved visual guidance of the hand to the target. So let's look at how this influences the reaction times. So let's look first at saccade reaction time. In both cases, you see that the reach is influencing the saccade, so you see the facilitation of the saccade for the SOAs that are closer to zero. For the reach, you see facilitation for these shorter SOAs only in the mutual excitation model. Obviously, in this case, the saccade is not influencing the reach at all. When we look at the reaction time correlations, we see for both models, there is a positive correlation for those shorter SOAs. And this makes sense because when both of them are being driven to their threshold at the same time, you're going to see that fluctuations in one will affect fluctuations in the other. So how do these actually fit our behavioral data that I showed you at the very beginning? So we fit these free parameters to the behavioral data of our monkeys, the saccade reaction time and the reaction time correlations. We did not fit these to reach reaction time. Remember, there were some other effects going on in the reach reaction times that made those somewhat complicated. When we looked at the best fit parameters, I want to draw your attention really just to the beta values here. This is how much each plan is potentially influencing the other. This is more like those models of asymmetric excitation, where the reach is influencing the saccade, but less so vice versa. Right? So as I said, this makes sense. The saccade, we're making saccades all the time. These are less likely to be, it's less likely that they would influence our reach than vice versa. So let me summarize this portion of the talk. So I've shown you that reaction time correlations decrease and the saccade reaction times increase with the increase in SOA. And when we looked at the various models that could explain the behavioral data, the model of asymmetric neutral excitation best captured that behavioral data. So obviously this has implications for how the brain might coordinate movements. So what might we then expect to see in the neural data? So let's think first about what movement plan should include. It should encode information about the location of the upcoming movement and also the timing of the upcoming movement. And based on what we saw in our modeling and our behavioral data, we think that the eye movement plan should be influenced by input from the reach plan. So thus we would expect to see the neural signals in any area that has activity related to saccade planning to encode the movement type, if it's a saccade alone versus a saccade with reach, as well as the location and timing. So now I'm going to move into the portion of the talk on neural data. So I was recording in posterior parietal cortex, and I want to justify why I'm looking at this area. So posterior parietal cortex includes area LIP, which is an area that has long been studied for its signals that are associated with saccade planning. It's also been looked at in decision making and a number of other things, but certainly it is involved in saccade planning. So this area also includes, the posterior parietal cortex also includes a nearby and highly interconnected area called the parietal reach region. And it's called the parietal reach region because it has activity that is related to reaches and reach planning. So neural activity in both of these areas has been shown to encode movements in common frames of reference, generally eye-centered frames of reference. And that could facilitate this interaction of the plans in both of these areas. And we also know that when there is damage to posterior parietal cortex, that this gives rise to sensory motor defects, including the inability to accurately reach to visual targets. So this is an MRI of monkey J. He's my favorite monkey. And so I've shown here where I recorded in area LIP in red. And then on the other side of that interparietal sulcus, you'll see in green the PRR recording sites. And I'm barely going to mention this, but as a control, we also looked at visual areas to see if we saw similar signals. When you want to look at planning activity, you don't want the monkey to move as soon as you cue the movement, right? And also, if you're going to look in an area like posterior parietal cortex where you see visual signals, you also don't want visual signals interfering with potentially recording information about a movement plan. Thus, we used a memory task, and I'm going to step through my task so you understand what the monkey is doing and thus the data I'm going to present. So in this task, again, he starts by touching a central green square and fixating a central red square. He then receives the target in one of 8 locations, and the yellow tells him he's going to make an eye movement and reach, with a reach. The target then disappears for a variable period of 1 to 1.5 seconds. And so we can look at activity during this planning period, which is no different visually from baseline, but now he has a plan in mind. He knows what movement he's going to make and where. Then, after that variable delay, the central fixation and touch points disappear. He makes his movement to the remembered location and receives his reward. So we're going to compare this to the saccade alone task. And in this case, the only difference is that the cue is red, and the monkeys are trained that means that they will make an eye movement alone. And so when he receives the cue to go, it's only the red fixation that disappears. He moves his eye to the target to receive his reward. Again, I didn't know exactly what kind of audience I was going to be talking to, so I'm going to carefully define some of these terms. So neurons tend to have, in this area, tend to have a preferred direction. So there's a response field, and when targets appear in that response field, you see a much greater level of neural activity. And when they appear in the opposite or null direction, you see much less activity. So after we map out the response field for neurons with the 8 different targets, we tend to present targets only in the preferred and null direction. And so the data I'm going to show you tends to be just in those two directions. I also, again, not knowing the audience, I want to very carefully introduce you to the types of neural data that I'm recording. So here are two example trials for targets in the preferred and null direction. And very often when you hear electrophysiology talks, I'm sure you hear about the spiking activity. So this is high-pass filtered data that just allows you to look at whether or not the neurons are spiking. You can set some threshold, and depending on whether you're looking at single cells or multi-unit activity, and look at the spike times. Also, there tends to be a lot of information in what's called the local field potential. So this is the oscillatory activity that the spiking activity sits on. And so we can low pass filter the data and look at this lower frequency activity. This is the local field potential. And you'll notice, so this is a single trial. I've shown where the target appears. I've shown where the go cue is given after that memory period, where the saccade begins and where the reach begins. And you'll notice that when the target appears in the preferred direction, there's much more spiking activity after the target onset that you don't see when it appears in the null direction. When you look at the local field potential, you'll see this oscillatory activity that changes with the target onset when it's in the preferred direction. You'll see this decrease in this lower frequency activity and an increase in this higher frequency oscillation. So we want to look at how is that local field potential changing across many trials and across different frequencies. So we can look at what's called a spectrogram. So we can look at what is the power at different frequencies. And now I'm going to show you data across this entire session. So these are many trials. And because of that, I've aligned things on the target onset and separately on the saccade onset because there's this variable time between these. And I want you to be able to look at the planning period and the early planning period and the late planning period. And this is, you'll see the frequency here, and what you're looking at is the change in power from baseline. So as you see in the single example, there's an increase in this higher frequency oscillation. You can see that here in red, and you don't see that in the null direction. Now this is all important. Does this make sense? Okay, good. Either you're all experts or I'm explaining it well. Either way, that works. So I want to look at how these local field potentials are affected by the task type. Are they encoding information about whether or not a reach is being added? So I've looked at the percentage of the local field potentials that are being suppressed or enhanced by the addition of the reach. So you'll see below the line in dark gray, the percentage of the local field potentials that show a decrease in activity at these various frequencies when a reach is added compared to the saccade alone. As expected during baseline, the monkey doesn't know what type of trial he's going to be doing, so there's no suppression or enhancement. But when the target appears, you see the suppression in the local field potential compared, when the reach is added compared to when there's a saccade alone. And the frequencies at which you see the suppression change through the course of the trial. So this is these are different epochs of the trial. When the target appears, this early memory period when the target has disappeared, the late memory period where you still see the suppression, the movement period, and post-movement. So the local field potential is encoding information about the type of trial. But what about the spiking activity, the single or multi-units that we're recording? When we look at all of the units that we recorded, we see that though it's encoding information about the direction, I should introduce what type of data I'm showing you. I'm showing you the firing rate, spikes per second. This is average across all of our cells, aligned on target onset and across time, and also aligned on the saccade start. So you can see that when the target appears in the preferred direction, there's an increase in the firing rate that persists through this memory period until he makes the movement. And you don't see that. If anything, you see a suppression in the null direction. However, when we looked at individual cells to see if they were encoding information about the movement type, we found a number that did. So we found those that seem to prefer saccade, or you can think of these also as having activity that's suppressed by a reach. So when you add the reach, you see less activity in these cells. You also have some, and this is about a third of the population, and about a third shows actually a preference for the reach and saccade task, and about a third show no preference. So we want to look at, is there something special about those cells that are encoding information about the task type? So here I'm going to introduce a new term, coherence. So when we look at the spiking activity and we look at the local field potential, these oscillations, they can be coherent or not coherent. When there's low coherence, the spike timing does not align at all with the phases of the oscillation. When there's a high level of coherence, the spiking activity tends to occur at a certain phase in these oscillations. Here I'm plotting the coherence across time and for these different types of cells that we've identified. Those that seem to prefer the saccade, those that seem to prefer the reach and saccade, and those that don't seem to have a preference. And we're looking at this in the two tasks, the saccade alone task and the reach and saccade task. And you'll see that there's a greater level of coherence during this memory period at these low frequencies, around 20 hertz, but only in those cells that we're calling saccade preference cells. So that means that they seem to have activity that's perhaps suppressed by the addition of the reach, just like what we saw in the local field potential. And these are the only ones that are coherent with that local field potential. So I've shown you that area LIP encodes movement type. The local field potential at these low frequencies is suppressed by the addition of the reach, and those cells that are coherent with that local field potential also seem to have activity that's suppressed with the addition of the reach. So next I want to show that the neural activity in this part of the brain encodes information about the timing of the movements. Here I'm dividing up the trials into fast and slow trials. I'm looking at how the local field potential across different frequencies changes as a function of whether or not the trials are fast or slow. And you'll see that when we look at this low frequency activity, there's an increase relative to baseline in the power in the local field potential, and that it seems to be suppressed when the reaction times are faster. And we don't see that, we see a hint of it in the null direction, but it doesn't reach significance. We see this to a much greater degree when we sort these trials according to the reach reaction time. So when he's faster, on the trials where he's faster to make the reach, we see less of an increase in this low frequency, or what I'm going to call beta band activity. And interestingly, we see this only when the monkey is making a coordinated movement. We don't see this in the saccade alone task. But I want to draw your attention to the fact that this increase in beta band activity is encoding information about the movement direction. So you see a much greater increase in this beta band local field potential activity when the target is in the preferred versus null direction. It's also encoding information about the trial type. So it seems as if this increase in the beta band activity is suppressed when the reach is added, and it's suppressed more for faster than slower reach reaction times. So when we compute a z-score to look at, in order to look at this encoding of reaction time across time of the trial, we see that the encoding increases throughout the course of the trial, and this is an area LIP. And I'm mostly talking in this talk about area LIP, but we were also recording neural activity in area PRR. We also see this encoding of reaction time in area PRR. However, we don't see it in the visual areas in which we recorded. So now I've shown you that local field potential encodes information about the reaction time. We also want to show that is there something special about those cells that are coherent with that activity? Remember, we showed that the cells that were coherent with that activity encoded information about the movement type. Here we show that they also are better at encoding movement direction, and in both tasks. So what about reaction time? So what about those cells that are, sorry, go ahead.
These cell preferences.
Yes.
Do we know anything, or do we know enough about the cells in these areas to know whether these are structural or functional properties?
No.
Of cells?
No, we don't. We do know that the cells that are coherent with the local field potential, we see a greater level of this suppression of the beta band activity. So it seems like maybe these cells are participating in these local circuits that are perhaps participating in this local field potential. The local field potential is thought to show us information about synaptic activity, and that activity could be from local circuits or it could be from incoming information. So you could think of it as potentially information that's incoming to area LIP that's giving information about the reach. But yeah, we don't know enough about the cells. Any other questions? Okay. So we wanted to look at whether or not the cells encoded information about reaction time. And we did an ANOVA analysis that showed that the cells that were coherent with this beta band activity and local field potential, they were more likely to encode reaction time. However, they did it in a way that varied, so it's not easy to show this in a figure. So we used a linear classifier to predict reaction time as fast or slow from each cell population, those that were coherent with the local field potential and those that weren't. And we see that during the reach and saccade task, those that are coherent with the local field potential are the ones that are also encoding information about the reaction time. And interestingly, again, this is only during the reach and saccade tasks. Those that are coherent with that local field potential, they are not showing, they're not encoding this reaction time during the saccade alone task. So to summarize the neural portion here, only the spiking activity, the cells in area LIP that are coherent with the local field potential seem to coordinate these coordinated movements. So the beta band local field potential activity in this area encodes the spatial location in terms of preferred versus null direction. This activity also encodes the timing of the movements, and interestingly enough, it's the greater beta band power that's associated with slower movements. So we think that perhaps this beta band activity is putting a brake on movement. And when you add the reach, remember during coordinated movements, the saccades are facilitated. So when you remove this brake, when you have less beta band activity, the saccades are faster. And we also see that in the reaction time. So, and the movement timing is predicted by the activity of only those LIP cells that are coherent with that local field potential activity. Thus, we think that the coordination involves these area LIP neurons that are coherent with the local field potential. So I'm going to sum it all up. I want to make sure that you're getting the main points. I know you've probably been drumming this drum for too long, but saccade and reaction times and reach reaction times are correlated during coordinated movements. We show through mathematical modeling of the behavior that these correlations seem to be due to interaction between the plans for these movements. And that we show through neural recordings that posterior parietal cortex, which always was a likely source of these interactions, it actually does seem to encode information about the plans, and that interaction between these areas may underlie coordination. And specifically, this increased beta band LFP in area LIP during the reach appears to influence the saccade timing through interaction only with those cells that are encoding the timing information about the movement. With that, I would like to acknowledge those I worked with. Bijan Pesram is a professor at NYU and was my advisor for this project. John Rinzel is also a professor at NYU, and he advised on the mathematical modeling, and his postdoc, Danny Marty, did a lot of that work. Maureen Hagan is a graduate student that worked with me on the neural recordings. Arpen Banerjee did a lot of mathematical modeling of the neural data that I didn't present here. Eva Su provided technical assistance. And the funding came from a variety of sources, which I very much appreciate. And thank you.